Hi, Victoria. How are you?
Good, thank you. Hi, Katarina.
Hi, everyone. Thanks for coming. We'll start in around 10 minutes. So, thank you. Hi, Z. Hi, Simone. We'll start in around 9 minutes. Thank you for coming. Nice seeing you here. Hi, Joyce. How are you?
Hi, I'm
Hey, everyone.
doing good, thanks.
Hi, Serena. How are you today?
Hello. Good. How are you? How is
Good.
everyone?
Thank you.
Yeah, hi, Serena, Victoria, and everyone else.
Hi, Joyce.
We'll start in around 7 minutes. Welcome, everyone. Thanks for coming. Thanks for being patient with us.
Hey Joyce, hey Serena. Sorry I was busy before.
Hello. How's it going?
Going well, thank you. I'm just making some dinner. Nice to see friends in the audience. W and Omar and Hector, Amelia, Brian, and Z. I'm making dinner and I burnt it, so I'm making it over. If I have to
what,
be truthful.
what, what's for dinner?
Well, I was sauteing some red peppers and onions,
Mm.
and I went to check some email.
You're so much better than me. I just scrap off whatever was burned and then give everyone.
It tastes good, but I'm so scared of that part. You know, I've heard so many times that we're not supposed to eat blackened things. So I'm just obsessive about scraping that part off.
Hi, Tutu. I hope I'm saying your name right. How are you? Thank you for coming.
Hi. Thank you. Yeah, thank you for inviting. Now it works.
Your mic
Okay.
works. This is fantastic. Hello, Choo Choo.
Hi.
Hello.
Hi.
Yeah, we're so glad you're here. Thank you for doing this, coming here on Clubhouse to speak with us here today. We are really honored. Thank you.
Yeah, this is fun. I'm looking forward to it.
Yeah, so in order for everyone to be really into this talk, please turn around 20 times in each direction until you're nauseated. I'm kidding. Don't do that. Please don't. I'm just kidding.
Because this is an interactive talk.
Yeah. No, just kidding.
Appreciate the.
Tutu, we'll start in around 3 minutes, if that's okay with you. We'll wait a little bit until people have time to arrive, and yeah, and we go from there.
Yeah,
So
sounds good.
we're joking about nausea, but it's, I think that our bodies forget pain while we're, after the experience, but then when you experience it again, you remember really well. And somehow nausea is just as debilitating as pain. And the times that I've experienced nausea beyond the flu, I'm thinking my first experience with extended nausea was when I was pregnant with my first child, and it was months of nausea. It was debilitating, demoralizing. I didn't know how I was going to make it through the day. And I'm so interested in this topic because of that, because we experience hardship, and I think it makes us more, it increases our compassion for what other people are suffering. So I knew that my nausea had, an end date. But for people who don't and have it for different reasons, my heart goes out to them. So I'm grateful for your research.
Yeah, me too. I'm rather in pain than nauseated. Like, it's the worst. I had the same with my pregnancy. It's like so nauseated. I couldn't keep anything, and not even water at some point. So I had to get transfusions every couple days, and it's just horrible. Couldn't do anything. So yeah, I really appreciate your work.
Yeah, thank you. I agree. Like, pregnancy-induced nausea is so prevalent. Every woman, or most women, may experience it, but we actually don't know much about that at all. So I'm surprised, but hopefully we can address some of the questions and in the current or future work. So yeah.
Well, it doesn't surprise me that not a lot is known about it, according to medical science anyway. Maybe traditional wisdom would have a lot to say about it. I know that I've read that it's that chronic gonadotropin is responsible, that the more of it that you show in your blood, the higher probability that you will experience nausea, which can indicate just a healthy pregnancy. But about the mechanism, or just that it can create a very sensitive stomach lining. But even that, why does that go toward nausea? And I'm sorry, Katarina. Yeah, I was threatened with the same, but I discovered watermelon. So now I have to say to anyone, if you're having trouble keeping water down, then try watermelon.
Well, that's an interesting one.
Yeah,
I'll
yeah, it kept me out of the hospital.
I love watermelon too.
Yes, and now you can buy personal watermelons.
Personal one.
Yeah, that's what they call them at the market. Little
Oh.
ones, they're fully ripe, but they call them personal watermelons.
Do they have personal kiwis?
Yes.
Okay, I think we can slowly start and then yeah, and go from there. So, welcome everyone to Science Society, and of course, a special warm welcome to you, Chuchu. We are very thankful that you came here today to share your really interesting and also very important research. So, before we start, let me give everyone a short introduction so they get to know you a little bit. Dr. Tutu Zhang, she's a research fellow in cell biology at Harvard Medical School, and she she did her PhD in neuroscience at the University of California in San Francisco, and she did her bachelor's in biochemistry at the Hong Kong University of Science and Technology, and she is as a neuroscientist, she's really interested in understanding how organisms detect and process different types of sensory stimuli at the secret and molecular level. And she's currently in Stephen Liberal's lab at Harvard. And there she wants to investigate how sensory signals from the gut are processed in the brainstem. And yeah, that's what we are learning here today. But before we start, Victoria usually asks a couple of interview questions, if that's okay with you, and then we go into your talk about your research. Thank you.
Yeah, sure.
All right, thank you so much, Chuchu. Thank you, Katarina. Chuchu, we want to make the most of your time here. And so to learn a little bit more about you before we hear about your research, my question is if you can think back in your childhood or early life and consider when you might have noticed that you had a special connection to science. That could be through an event or a person or an experience.
Yeah. So my father is a scientist, and so I grew up in a university campus pretty much, and I often go to his lab when I was a child, and so for me, it's always kind of part of my life. And then when I went to college, I of course thought about this kind of research life. So I tried it. I joined a research lab to be like a summer student, and then I think I really connect with the people in the lab. I think my mentor, also the people that I work with, they're just really fun, smart people, and I really like being with these people. So I think perhaps it's the people and of course the exciting science that really connect me with this path. So I basically decided to pursue PhD. Then I can keep doing what I like to do. So this is just how it started for me.
Thank you very much. That is really interesting that the roots of that began in your father's lab. And so were you listening, or was this was this lecture, or you were observing him with research, or manipulating data, or what types of um, lab activities were you witness to?
Yeah, so he does different things. He's a material scientist. So I go to his research lab where there's some like super hot ovens that they make certain materials that I actually don't quite understand. I just kind of see it, see the people in the lab doing stuff. I don't understand. I basically just like observes. Um, and then yes, sometimes my father took me out of daycare. Like after my daycare finished, he basically took me to the lab because he still have to finish it up. So I basically just sit there, watch what people were doing, and then go home. So yeah, I never go to his lectures. That's too much.
You just got the fun part. That sounds so fun. You got the best field trip ever, and it was your dad. So hooray for him. That's really beautiful. And then can you take us up to your current research? What events, maybe just a broad overview or brief overview of what led to the current research that you'll be presenting this evening?
Yeah, sure. So I've always been interested in sensory neuroscience. So as an undergrad and then graduate school, I always studied like sensory processing. So when I was a grad student, I work in pain, and I think some of you mentioned about like how terrible pains are, and so that's what I studied as a grad student. So I basically identified pain-producing animal toxins from snakes or centipedes or scorpions, and use them to elucidate mechanisms of pain. And after I finished working on my project, I started to think about so pain basically is a sensory neurons detecting external dangers such as toxins. So what about toxins that are ingested, so internal toxins? And this happens a lot when we ingest pathogens and cause food poisoning and nausea and vomiting. And then I start to think that like nausea and it's a very similar protective sensory mechanisms to avoid dangers, but not much is known at all about nausea. So then I joined my current postdoc lab, who is study the internal sensations, and we use a lot of current neuroscience techniques to study how bodies detect internal stimuli, such as ingesting toxins like stomach pain, these kind of things. So that's kind of how I get to the nausea field.
Right. Thank you. Yes, it is so fascinating that we have the unpleasant feelings of nausea and pain, and yeah, everything that we just said. Going back to our just discussion about how debilitating that can be, and what is the protective mechanism? What could those possibly be? So at this point, you're welcome to go into your body of your discussion, and we can have a Q&A following, or we can have the Q&A drive your discussion along, either way. And sometimes friends will put questions or comments in the chat, and we on stage will take care of that for you, so you don't have to worry about it, and we'll share that with you. So we can run this however you'd like, and the floor is yours. And thank you, Chi.
Yeah, so I think I will talk about our two recent studies as they're closely connected. I will talk about the main takeaways in each study, and then I can open up for questions. And of course, you can always interrupt me during the talk for any questions or clarifications. So, so nausea is a sensation of visceral malaise, frequently associated with vomiting. At some point, everyone experiences nausea, whether it's from food poisoning or it's through infection. Nausea and vomiting is evolutionarily conserved protective mechanisms to get rid of ingested toxins, and also learned to avoid these ingested toxins for the future. Most of the time, it's only temporary, but for some people, such as those on chemotherapy treatments, nausea can be severe, chronic, and even life-threatening when it prevents treatments and cause dangerous dehydration. The sensory pathways leading to nausea have remained largely mysterious, and particularly at the molecular and genetic level. So this led to our first study that we want to identify and characterize neurons that regulate nausea-like responses in mice. We started focus on a brain structure called the air postrema. It's located in the brainstem. The air postrema is long been associated with vomiting, and is one of the rare parts of the brain that's outside the blood-brain barrier that can protect the brain from the exposure to bloodborne toxins. So for long, people thought that the air postrema role is is an alarm bell for dangers. So there are different cell types and receptors expressed in the air postrema, but we don't really know what they are. So to start, we built an atlas of cell types by characterizing gene expressions in the thousands of individual neurons in the air postrema for mice. So this is by a technique called single nucleus RNA sequencing. The idea is that different cells playing different roles will express different genes. And based on how similar the functional gene expressions are, we can group cells, and supposedly each group will be doing something different from one another. So for example, excitatory neurons and inhibitory neurons are separate groups because one elicits excitatory responses while the other elicits inhibitory responses. This is a well-established technique in recent years by many research groups. So what we found is that there are only a handful of different excitatory and inhibitory neuron types in the air postrema. Of particular interest were excitatory neurons that express glucagon-like peptide 1 receptors, or GLP-1R. It's a receptor on the cell surface that has linked to blood sugar and appetite control. In fact, agonist for GLP-1R is the mainstay for diabetes treatment, but induces nausea as a major side effect. To probe if these neurons play a role in nausea, we used a conditioned flavor avoidance behavioral test in mice, as mice don't vomit unlike humans. We add cherry or grape flavor to the animal's drinking water, and then gave the mice either an innocuous substance or one known to induce nausea. If a mice feels malaise, it will quickly associate a food flavor with the negative sensation and avoid it in the future, similar to that you would avoid a specific food that makes you throw up. We tested many different substances, including gut irritant, lithium chloride, bacterial lipopolysaccharide associated with food poisoning, diabetes drug, GLP-1R agonist, and cancer chemotherapy drug, cisplatin. As expected, all tested substances will lead to a strong flavor aversion in mice. When the air postrema GLP-1R neurons were experimentally removed with a genetic approach, mice stopped developing flavor aversions for most of the substances, suggesting that they no longer experienced malaise as normal. We also experimentally turned on the air postrema GLP-1R neurons using a technique that essentially tricked them to think that there was a toxin present. Mice with activated GLP-1R neurons would acquire strong flavor aversions even when they were never exposed to a nausea-inducing substance. So both loss of function and gain of function of these neurons and observations that we saw suggest a connection between these neurons and the nausea responses. There are some other things that we also discovered. The air postrema GLP-1R neurons can be further divided into smaller subsets. One subset express a receptor for the stress response cytokine, GDF15. And interestingly, silencing this subset of neurons will cause the mice to stop developing flavor aversion for only the lithium chloride-induced aversion, indicating there's potential a division of labor between different air postrema neurons with different neurotypes responsible for different alarm signals for like ingested toxins. We also mapped neuronal connections of the air postrema GLP-1 neurons in the brain. These neurons connect to many other regions of the brain, including one called the parabrachial nucleus, which has been recognized as a sensory hub for pain and aversion. So this may be how the air postrema neurons help induce conditioned aversion memories. So this is the brief sum up of the first study. And what inspired our second study is that when we mapped the air postrema neuronal connections, we also look at the air postrema inhibitory neurons. These neurons, surprisingly, only project locally within the air postrema, forming a dense network that connects to the nearby excitatory neurons. So we thought, maybe the inhibitory neurons can stop the excitatory neurons that produces nausea. Indeed, when we activated these inhibitory neurons, mice lacked nausea-like behaviors that are typically caused by excitatory neurons. In our previous sequencing results, we also identified 3 types of inhibitory neurons in the air postrema. One of these types expressed a receptor for GIP, which is a gut hormone released after eating that stimulates the release of insulin to control blood sugar. We were curious whether this population of inhibitory neurons could be manipulated to suppress nausea behaviors, and if so, how does it work? When we used GIP to activate these inhibitory neurons, the inhibitory currents prompted by the neural messenger GABA flow to nearby excitatory neurons, which then, and actually interestingly, specifically to one subset of the excitatory neurons marked by the GDF15 receptor that induces nausea, and then reduce the overall activity of these neurons. On the behavioral level, we give mice flavored water again, followed by injection of nausea-inducing substance, and the animals will begin to avoid the flavored water. But if mice receive GIP, the rodents were happy to drink the flavored water. But if we bred the mice that lack the inhibitory neurons in the air postrema, giving GIP to the mice will make no difference. The animals will continue to drink, avoid the flavored water. So GIP indeed suppresses nausea-like behaviors, and it acts through the air postrema inhibitory neurons. So GIP has already been studied as a potential treatment for nausea, but people don't understand why. And our study basically suggests that it's through the air postrema inhibitory neurons. And for those other studies, they have actually shown that GIP, when given to animals that do vomit, including ferrets, dogs, and shrews, it can reduce nausea in several nausea models, such as cancer chemotherapy-induced nausea and some other conditions. And there are also people working on incorporating GIP into diabetes treatment that target the GLP-1R receptors with the goal of reducing nausea as a side effect. So the traditional approach to intervene nausea is to block signaling pathways using pharmacological inhibitors. But we here focus on a new strategy of using the inhibitory neurons that suppress the excitatory neurons in the air postrema. By identifying the inhibitory neurons that suppress nausea in a pharmacologically accessible way, we can simply engage this neuron to counteract the nausea responses. So this is the main takeaway of the paper, and there's a lot of interesting questions that I think opened up, and I I'm currently actually searching for faculty positions where I can establish my own research groups to study nausea, and I hope to study some of the interesting questions um, that I will be discussing. For example, one I'm really interested is to understand the way the gut communicate with the neurons in the air postrema. So the air postrema excitatory and inhibitory neurons are connected to the gut through the vagus nerve. But how those brain cells are controlled by gut-derived signals are unknown. Obviously, we know that when something goes wrong in the gut, you can induce nausea. But how does that work? And maybe some of these mechanisms are conserved in certain gastrointestinal disorders, such as food allergy. And another question I'm also very interested is to uncover the potential different signaling factors that contribute to nausea. For example, food poisoning potentially engages signaling molecules and cells very distinct from cancer chemotherapy or pregnancy-induced nausea. I'm particularly interested in many of the pathological conditions that induce nausea without the presence of even a foreign substance, such as during radiation exposure, pregnancy, and migraine. So nausea and vomiting in these conditions no longer serve as a protective reflex, but become a prolonged symptoms that reduce the quality of life. The mechanisms behind this sort of maladaptive nausea conditions are complete mysteries. Presumably, our own cells within the body start to secrete sensory cues that stimulate nausea responses. So what are those signal cues? What cells generate these maladaptive signals? And how are the signals detected by what cells? So we want to know more about the various nausea mechanisms so that we can develop even better treatment strategies that can tailor to specific conditions. So to that, and from there, I think I can open up to questions.
Yeah, thank you so much for giving us an overview of your really interesting research here. And yeah, I agree that your research opens up a lot of questions, like every really great research project does. It just opens up basically a new field almost that you can probably have material for 3 professor like assistant professor positions, like PI positions. So and we wish you all the luck, of course, for that, and that you find the perfect place for you. Okay, so going into questions, so are you maybe currently working on maybe dissecting out also if so you talked about vulnerabilities, right? But before that, do you are also like maybe some receptors, GABA receptors, also involved? Like, is there any data that just blocking, like using maybe epilepsy drugs could also since inhibitory neurons are also involved, could that be like an immediate target that one could use also for nausea suppression maybe?
Yeah, so I think use the generic GABAergic receptor manipulation could be difficult because they're too non-specific, and there are, it could have multiple other effects. And so I think the idea is that we now know that these inhibitory neurons could potentially suppress nausea on the circuit level. So we can look at what receptors these cells express. For example, they're often GPCRs or receptors that we have known targets, and we can manipulate those neurons much more specifically with those receptors. And I think this might be this may have already been a strategy utilized by companies to develop new anti-nausea drugs. So I think also, in a way, we can also look at excitatory neurons in their postrema. What receptors do they express? And in our paper, we actually showed that by looking at the receptors in the excitatory neurons that produce nausea, we can actually test. And indeed, those signaling pathways engaging excitatory neurons induce similar effects like GLP-1 agonists, like GD15 receptor. So basically, it's very powerful to actually just look at the gene expressions and look into the signal mechanism engaged by different subtypes of the neurons and develop better pharmacological tools.
So the brain cells that you discovered, are they responsible also for like vomit inducing type of behavior, and so contractions in the stomach and things like that? Like, did you measure this type of control, motor control or so in the animals?
Yeah, great question. So, yes, I measured physiological changes, including breathing, heart rate, gastrointestinal, gastric pressure, so pressure within the stomach. And when we activate these neurons, we observe a increase of gastric pressure, so basically stomach contraction, and a decrease of phasic pressure. So we know that stomach contract at a regular pace. When we activate these neurons, basically the regular contraction would decrease. And this is indeed consistent with when we feel nausea and feel stomach malaise. However, it's a little tricky because mice don't have a vomiting reflex, so it's very hard to say, this is similar to nausea, vomiting, because they don't have vomiting reflex. But I think you're asking a great question because it's very important to measure in a vomiting animal model what happens if we manipulate these neurons. And there are some research group that actually did use vomiting models. And I think in my future lab, I would also like to incorporate that into my own research and to see what happens when we manipulate these neurons.
Yeah, that's very important. Thank you for that. Because that it's more personal. I have close family and friends in North Portugal. There's a high occurrence of ALS, like a specific types of ALS, and also muscle dystrophy. And a big problem is nausea and kind of vomiting inducing. And it would be really interesting if maybe by inhibiting that brain region, if you could basically reduce those relatively early onset symptoms. So yeah, so if you would one day also study muscle dystrophy animals, that would be really, really interesting. Thank you.
I don't know if you said this, if I might have missed it, but do you think this might also be involved in the nausea that occurs with migraines? Sometimes it's even without the pain of the head, basically, but it's kind of an abdominal migraine. And I've had a few migraines in the past where the nausea was the worst thing. Thanks, I'm done.
I'm sorry to hear that. Um, so I honestly, I don't know, but I think it would be very, very interesting to look into that. Migraine, there's still a lot unknown about what causes migraine. So, and the animal model for studying migraine is also very difficult because you can't ask mice whether they're experiencing migraine. So, but there are quite a lot of research groups working on migraine, and in the future, I would definitely like to join force with people who are expert in migraine and look into that. I, just kind of thinking about migraine, some people there's studies suggest that CGRP is very important for migraine mechanisms, and I wonder if some of this kind of signaling molecules also can be detected by these neurons. Currently, I don't know, but I think it's a very great future questions.
I was also thinking of the connection with the gut because quite a few people report having foods apparently triggering their migraines. And I know there's some ideas about mechanisms, but I don't think it's all explained what the mechanisms are. Anyway, I'm done.
Wow, very interesting. I have not heard of that, but this is super interesting to look into. Mm, yeah, yeah.
I mean, I'm curious about, so a couple of questions in terms of getting to human trials, or, at least looking at the fidelity of the mouse model in extrapolating to humans. Has there been much work on that? And is it a really good model in the sense that we can sort of design very specific therapies?
So I think the question is whether mouse model is a good model for studying nausea. Um, I think it's not perfect. For sure,
Mm.
it's not perfect. I think you always need to have a vomiting model to validate. So, such as ferrets, it's a very common vomiting model used by preclinical studies, and um, the this assay that we do called conditioned flavor avoidance assay is suggestive of nausea, but it's not definitive, and you do need a vomiting model to do it. So most of the translational work I've seen is to do mice and then do ferret models, and then we can move on to human trials.
And I wonder if is there an indication that the effects of the malaise and the just awful feeling is somehow separable from the vomit reflex, in the sense that if in the cases of food poisoning or something, where you really need to get it out, uh, versus okay, can we damp the malaise and still have the signal that something's wrong, but it's just not so awful.
Um, yeah, so that's also a great question whether malaise or nausea and vomiting can be separated. In a way, I would. So there's suggestions that it indeed can be separated because a lot of the drugs, current antiemetic drugs, are relatively efficient in. Well, they're not very good, first of all, but some of them are more efficient at stop vomiting, but not really treating nausea. So people think that maybe those drugs targeted the vomiting region, but not really suppressing the sensation. Um, I think the, the vomiting is a motor reflex. So anything that so you need so basically sensory cells detecting signals, and then you will go to some interneurons, and those interneurons will eventually go to the motor neurons to trigger the vomiting reflex. And the sensory neurons will also send information to the higher brain regions to cause malaise and learned aversion. So the postrema neurons that we studied are these kind of sensory neurons that can both send information to the higher brain to condition aversion, but also send information directly to motor neurons to cause different sort of reflexes, such as the GI contractions that we measured. So I think you can target different pathways, outputs from these neurons to sort of separate them. I guess so. That's my answers to your question.
There's been some times with food poisoning, it would have been nice for someone to just come in with a gamma knife and just. Are there regions of neurons that might be? You said it was the same neurons that are sending the signals to the brain as they are to the motor neurons. There wasn't a subpopulation that was identified that could be targeted?
We don't know. We don't know. Yeah. So um, even though we know the cells can directly send motor outputs to control, stomach contraction, we believe those motor neurons could also be modulated by surrounding interneurons, as the brainstem is composed of other nucleus. So there might be a unidentified population of interneurons that could modulate the reflex itself. Yeah, I think it's very likely, but we haven't we haven't done that.
Thank you. Fascinating.
Yeah, thank you so much for fascinating work that you just shared with us. As you mentioned, that one of the side effects after receiving the chemotherapy for the patient is the nausea, and you might know that with the immunotherapy, somehow, especially using of the, checkpoint inhibitor, solve this problem. However, because you just mentioned about the GLP-1. I was just wondering if you want to use this mechanism, what about, some of the patients that they are suffering from the pancreatic cancer? Is that possible that reversely you exacerbate the situation with developing the drug or such things based upon your research or not?
That's a great point. I think we have not looked at like pancreatic cancer model in combination with of this GIP drug, and I think to think about the translational potential of using GIP to treat cancer chemotherapy-induced nausea, it definitely have to be more well-rounded to think about other potential issues like the one that you raised. I would think that at this point, GIP is more thought of a potential target for treating diabetes diabetes drugs induced nausea, and because it actually worked together with GLP-1R agonists to actually increase the efficacy of blood glucose lowering and as well as weight loss, increased weight loss. So for that application, I think it's very promising. But like you said, for other nausea conditions, this may not be the perfect drug, and this is also promotes one of my research goals, which is to study even more broadly what signaling mechanisms are involved in different conditions, such as cancer chemotherapy. Can we even have a better target than just blocking with the GIP receptors or activating GIPR? Yeah.
Also, do you have any thoughts around the plasma instability that it can happen because of the using the substances or somehow chemotherapy or such a things? I was wondering, knowing your thoughts around that.
So sorry, ma'am, if am I understand your question correctly? Is the stability of the
Plasma,
GIP plus
yes,
GIP?
plasma instability.
Yeah. So yeah, that's so GIP and like you said, GIP and GLP-1, they're both very unstable. They're gut peptides that are secreted and potentially acted relatively locally, and they don't really circulate that easily. But we think that in certain conditions where there is potential large secretions, maybe they could actually target the cells in the air postrema. But we also have to consider the pharmacological or therapeutical use of these drugs. Usually, people choose to use a stable analog of GIP or stable analog of GLP-1, so they actually stay quite long in the circulation. And this is actually what we use in our study as well.
Thank you.
Hi Sterling, how are you? Did you want to ask a question? And Denise, did you want to ask a question? Sorry,
It's
Okay.
okay. I'll go after Sterling.
Okay, thank you.
Okay, thank you. Great talk, great paper. I actually saw this on Twitter when it came out, so it's cool to just get invited here. So just briefly, my area that I work in a lot is in vagus nerve stimulation through like cervical stimulation through ultrasound, so sonogenetic kind of stimulation work. And there was a question like Serena had asked about, I think, which a couple questions about migraines related to these kind of networks. And um, there are existing tools available on the market that do stimulate the vagus nerve through electrical stimulation called the gamma core. That's from a company called Electrocore. So there are migraine-based treatments that may also, stimulate the same network in the AP and the vagus nerve. I was curious for the speaker today, what kind of so you did optogenetic stimulation. Have you tried, I guess, what kind of experience do you have with like direct vagal stimulation? Could you have gotten the same effects if you did if you stimulated the nodose ganglion? Like how far out of the area postrema did you try optogenetic stimulation to try and elucidate that network? Did you go any further out of there with your work?
So, first of all, I have not stimulated nodules and look at the sort of behaviors, but I think there's a couple of things. Our lab studies the vagus nerve, and the lab has discovered that the vagus nerve is composed of the genetically and functionally different subsets. Some vagal neurons mediates detects a stomach stretch, some detect stomach hormones, some detect lung inflation, some detect heart rate. So they're all different roles collectively together into those nodules. When you stimulate the nodules all together, it's sort of there's not too much specificity, and so that's the reason that I don't do this kind of study. However, I do find that there are specific vagal types that do project to the air postrema, but we don't know what those neurons do. Do those neurons innervate the stomach or innervate the duodenum in the intestine or the colon of the intestine? We don't know. And if you stimulate those neurons specifically instead of the whole vagal nerve, maybe you could actually get the same sort of aversion, like nausea-like responses. So and if you stimulate like everything together, some of the effect can be potentially masked by some of this other very strong effects, such as heart rate or like stop breathing. So I think it's a little bit difficult to stimulate the nerve in experimental conditions. Yeah.
Cool, thank you. Yeah, that answers my question.
Go ahead.
So I had a sort of more basic question. There are some psychologists which assert that pain can be thought away. What do you think about that?
So it's a question that nausea can also be thought away. So that's a question of how do nausea or those neurons that we discovered may be modulated by higher brain inputs, and I think that's a very interesting direction as well. I did not mention that, but I think those neurons also receive inputs from higher brain areas. And if you, in your own experience, maybe you meditate, you can kind of suppress your nausea to somewhat. Or if you see something super disturbing, you can feel nausea sometimes too. So definitely, you can use the higher brain to either invoke or suppress nausea. So what are those neuron pathways? We don't know. So we can potentially trace neurons from where what we have discovered to the higher brains and see, what kind of inputs do they receive, and kind of maybe go into that question a bit more. But at this moment, we have not done that. Yeah, it's very interesting.
What would it take to do that?
So it's not tricky actually. It's potentially a quick one-step experiment. So viral tracing is very commonly used in neuroscience, and basically you just need a virus that can trace backwards to to identify the inputs of those nausea-inducing neurons. And then another potential way is to look at the inhibitory neurons that were discovered. One potential way that we might suppress nausea is by activating the inhibitory neurons, right? So maybe those inhibitory neurons receives higher brain inputs. That's how it suppressed the nausea. So yeah, we just need to carry out some tracing experiments.
Fascinating.
Can I
I'll be very interested to read those findings when they do come out.
can I
Thank
jump
you.
in real quick? I just want to can I think this is fascinating. That's a great question, Denise. The one area that I've been hearing a lot about is the insular cortex and how it has a direct projection to the area postrema and the PAG, the periaqueductal gray, and that area in the insula does have projections back and forth between the prefrontal cortex and then the immune system and branches of the vagus nerve. So you can have insula like invokes emotion and thoughts and memories, and so I could see that. Yeah, I don't know what exact neuronal networks there would be, but I think that there could be something to that for sure. Obviously, it'd be hard to do that research in a in a fly brain, or but maybe with mice you could condition like some trauma response and then try to evoke different kinds of memories that could suppress that network. Just an idea, just because that I wanted to throw that out there. It's a great question. Thank you.
Yeah, great comments from Sterling. If I were to bet where that could be, I'd probably bet it's the insulin cortex. If I trace back the air postrema neurons. So, and I think yeah, if I were to do this project, I would starting with a mice genetic model to trace back those nausea inducing and suppressing neurons in the air postrema to see where do they receive inputs, and then we can start from there.
Yeah, just wanted to mention, like in your paper, you showed that by using agonists of the GIPR receptor, you could reduce this cancer drug-induced vomiting, or it was reported before that in 2018 that this cancer drugs-induced vomiting in ferrets could be achieved, which is really interesting. How specific are these receptors to these brain regions, or are they quite abundant around the brain?
Yeah, so there are other regions expressing GIP receptors, and for example, in the hypothalamus, where it plays a role in feeding. Um, so, but in our paper, we show that if you ablate the air postrema GIPR neurons, then the malaise or the protective effect of GIP is lost. So even though in other brain regions they still express GIP receptors, those are probably not responsible for the anti-nausea effect. So yeah, so even though it might have broader effects than just anti-nausea, yeah.
That's interesting. And nausea can induce PTSD-like symptoms in animal models quite easily, right? It's another way of inducing this type of mechanism of like ingrained memory, like kind of negative labeled ingrained memory. It's very efficient. So because Denise asked about, you know, controlling with the mind, basically the nausea. Do you think that insight from PTSD like memories in animals could give insights in how to control nausea? Like, is, or do you think that's a very different mechanism?
Um, I'm not quite sure if this is the same mechanism with PTSD-like mechanisms. I do know that the pain and nausea likely go through a similar pathways through the parabrachial nucleus, and then to the central amygdala to condition the aversion and the all the negative memories. So, and this learning pathway could be relearned or it's plastic. So after certain so once you receive the nausea stimulus, but if you don't give the nausea stimulus and kind of conditioned the same flavor but without the presence of the nausea stimulus, mice actually also forget. So they're eventually okay with the flavored water that we give them. So I wonder if this sounds like it's a similar pathway. Maybe you study that PTSD. You can tell me
Yeah,
more.
like I yeah I in Joseph Ledoux lab I studied fear memory, but a friend of mine, Hillary, um she went to Cold Spring Harbor and started actually gustatory memories, and you know, one way of having very negative gustatory memories is inducing, like nausea inducing, like mixing that into the food and then having basically the same type of mechanism, just with, you know, a gustatory input instead of you know, the noise and stuff. So it's very effective and very fast mechanism of inducing like negative labeled memories. So yeah, I was just wondering if then the, you know, controlling basically the nausea could, you know, what you described is basically extinction training. Um, if there are similar, you know, receptors involved, is norepinephrine and stuff involved, and if you could basically use that stuff to also like control nausea, and would be, it would just be really interesting if both of those are kind of really similar. And it would make a lot of sense because it's a huge threat, right, to eat something poisonous, and you should learn it fast. The same as if like a predator comes and tries to hunt you, you have to really learn it fast. So it's really important for survival. So it would make really sense if there are similar mechanisms involved. Yeah, thank you.
Yeah, thank you. Makes sense. I'll look into that. Yeah.
I see Susan. Hi, Susan. How are you today? Do you have a question?
Yeah, I had to leave, so I'm not sure if you covered it. I have two quick thoughts, and hopefully they would lead to a question. One is, if you had not discussed it, the correlation between nausea and motion sickness, and if there was anything in your research there, and why some doctors prescribe Benadryl for nausea and why it seems to be so effective. Again, this is not my field, so these are just, I'm curious about both things. Thank you.
Yes, I did not talk about motion sickness. Great question to have a chance to discuss that. So motion sickness is thought to be mediated through the inner ear, the vestibular system. It's a completely different sensory pathway, but it has been shown, or there's some evidence showing that the vestibular sensory pathway do also converge onto the parabrachial nucleus for the aversive effect, similar to with postrema neurons project to. So potentially, these are two different sensory pathways. One detecting bloodborne ingested toxins, one detecting motion, and both pathways can converge onto a nucleus in the midbrain to condition aversion and induce nausea. So, and the antihistamines are used to treat motion sickness because it has been shown that the vestibular nucleus express the histamine receptor. So it's more kind of specific to that particular pathway, but not for the the air postrema potential the pathway.
Just a quick follow up. Had you read anything about motion sickness and nausea in relation to people that are highly creative being more susceptible to those things?
Oh, interesting. Oh, I have not heard of it. Yeah.
I've
Okay.
actually read something. If I find it, I will send it to you.
Okay. Yeah.
Okay, thank you. Thank you for answering.
Could I ask real quick, just to jump in on that the comment about Benadryl? That's something that we know in our work as an anticholinergic. So it does seem to block acetylcholine at the site of the receptor. Did you look at cholinergic knockout in any way in this area postrema in your work?
No. So we look at a bunch of receptors that are common anti-nausea drug targets, and for example, serotonin receptors and NK1 receptors and histamine receptors. So the air postrema is not rich in any of those receptors. It does express some of the acetylcholine receptors, but we have not looked at the like knockouts, like you said. But interestingly, air postrema receives dense vagal inputs, and the vagus nerve is rich in expression of serotonin receptors and K1 receptors. So we think that a lot of the anti-nausea drug potentially are inhibiting this vagal air postrema communication, and that's perhaps how it does, how those drugs work at the brainstem level. But we're actually actively testing those theories. Yeah.
That's interesting. Just as a follow-up, I don't want to take up too much time, but a question would be if you think about the feedback loop, right? So you eat something that makes you sick, the signals go up your vagus nerve into the area postrema, it detects a certain threshold of activity, and then the area postrema induces like vomiting by sending signals down through the efferent pathway to induce, the gag reflex and to vomit. Um, okay, if you take an antiemetic drug like I don't know what I don't know so if you have any examples of I think
Densitron?
there's on Dancitron, yeah, exactly. So if you take that, is it what is the mechanism where it's actually activating in that feedback loop? Is it simply stopping the vomiting response, or is it more stopping the nausea responses from reaching the area postrema, or is it knocking out both?
Wow, you're asking the exact questions that we want to figure
Okay.
out. So we don't know. So the drug has been used for so long, and we know it's a serotonin receptor antagonist, but we don't know where it acts and how does it prevent nausea. And so we want to. I mean, I want to look into how exactly it works. Like, is it stopping the inputs from vagal to apostrema, so it kind of stopped the whole circuits at the beginning, or it stops the some of the intermediate levels, the vagal to motor neurons or something. So we don't know. Yeah.
Thank you.
I just want to take a moment to pause and reflect on the fact that isn't that interesting? Like it's so commonly used, but we don't actually really understand what's happening.
Oh yes. The nausea, whole nausea field. I feel like there's so many unknowns. The drugs were used, but we don't know how those drugs works, and it's so common, but people just don't know why, how. So I am so excited to study it, but there are just so many questions. Yeah.
And it also turns out so many drugs have so many different mechanisms of action. It seems like, I've Googled this with regard to a lot of antibiotics, and I found out a lot of antibiotics that are known to be used for bacteria maybe are also antifungal, or they have immunosuppressive effects, or so on and so on. Anyway, I'm done.
Yeah, and also another point is that the drugs are not perfect, and the drugs work for some people but not for other people, work for some condition but not the other condition. So it's we really need to understand the mechanisms of nausea so that we can actually develop better drugs. And yeah, that's the goal of the research.
Uh, could you elucidate? So based on your optogenetic work, is it how much selectivity do you have in the directionality of blocking effects? So, because the AP takes afferent inputs from the stomach through the vagus nerve, so there's afferent nerves that you could probably um, cut off somehow or like selectively turn off, and then you could potentially turn off the mechanisms of AP sending, because there's a conscious feeling of feeling sick, right? Which is definitely a projection towards the conscious mind through the insula to the prefrontal cortex for the behavioral changes. How specific do you think you could get in the next few years to knocking out either one direction or the other?
So one, we did this experiment to trace whether the motor projecting uh, uh, terminals are the same as are from the same neuron as the higher brain projecting axons. So basically, we're tracing at two brain sites, one in the higher brain and one from the motor nucleus, and going back to the neurons that have the axons, and to see whether it's the same neuron or different neurons. And we actually found that most of them are the same neuron. So basically, one postrema neuron will send axons both upstream to the higher brain and downstream to the motor reflex. So it would be very difficult to. You would think that if there are two different neurons populations, then you can kind of selectively block. But if it's the same neuron sending to both pathways, you, well, I don't know, but there might be ways. But at this moment I couldn't think of a good way to stop one but not stop another.
Interesting. Thank you.
Can I ask another quick question? Have you looked into the connection, or maybe you covered this already, between nausea and dehydration?
So dehydration is, I think, caused by throwing up too much, and you cannot keep anything down. And with time, then you would lose a lot of the water. Um, yeah, I think the main it's the fact that you cannot drink or eat, and you keep throwing stuff up. Yeah. Does that answer your question?
Yeah, I'm curious. Obviously everything you're doing is intent on treatment and helping people. This would be a really great way to understand how to help people in this situation, like people that are dealing with like a high fever, obviously during all these pandemics and viruses and infections, and then consequently being unable to control their nausea in relation to that would be. And sometimes it seems like if you can't control it right away, it's hard to then control. So it would be interesting to know, I guess, futuristically.
Right. If you can feel better by taking some pills, and you don't feel nausea, and you can drink water, you probably, yeah. I think that would be definitely the goals, right? For the for anti-nausea treatments, yeah.
Are there any common items that that people might be able to use that are not pharmaceutical? I remember when I was young, if I had those sorts of problems, family members would give me mint tea with lemon. Is there anything else that might help with this nausea state?
Um, I also have something like that that I do use, like for example, ginger. But I'm not gonna say that any of this have like scientific base. I have not studied that, and I would like to find some scientific base for some of the common wisdoms. For example, ginger. People say ginger works, but some people also say it doesn't work. So like, does ginger the ingredients in the ginger activate some receptors or suppress some receptors in the air postrema or the vagus nerve, that so that we actually have a real scientific base behind it? We actually don't know, but I think it could be studied, in the future.
Those are some studies I'd like to see.
Yeah.
I mean, like we were just talking about, it's not there's not any less evidence base than for some pharmaceutical interventions. We don't
Yeah.
know. No one studied it.
Yeah.
Kyle has a question. He wrote me about using cab- um, cannabis for regulation of nausea and vomiting, and it's also prescribed for people with undergoing cancer
Cancer.
treatments. So, did you study that in your work, like what the mechanisms are and where it would help?
Yes. So we have not studied it, and I think cannabinoid receptor is like any other anti-nausea drugs. It works, but we don't know how it works. But it's also very complex because um, over overuse of the weed also cause chronic nausea. So their receptors, this receptor are expressed in the air postrema, but it's also expressed in the GI tract. It's quite broadly expressed. So how does those different neural pathways engage at different dose at different time, and lead to the effects, either anti-nausea or chronic nausea? It's really not clear. Um, yeah, it's a mystery as well. I think it deserves a lot of future work on that.
Do you mean the broad expression as in the CB1 or CB2 or both receptors?
I think CB1 receptor. Um, I have to look into my expression of the genes, but I do remember CB1 is broadly expressed in the air postrema, and um, some of my colleagues studied the GI tract, sensory neurons in the GI tract, also see some um, the receptors. I think it's also CB1, but I have to check with him.
Another question I received is, so you basically think that nausea is something that is experienced in the brain, or is nausea experienced basically in the rest of the body due to what these cells signal to, the muscles and the whole, like, where do you think is it experienced, basically, that horrible feeling of nausea? Or because I thought this question that I received was really interesting, because if you think of it, if you could just disrupt maybe in the spinal cord the signaling through some, magnets, um, some stimuli, there are so many devices right now out there where you can like stimulate your body, your muscles, whatever. If you could disrupt that shortly for like severe onset of nausea, would that help? Or do you
Yeah,
think?
yeah. So you're asking a great question that I would like to study in the future. So what are the signals of nausea, and where do they get detected by what cells? And we know some of the neural pathways of a nausea, for example, the air postrema and potentially the vagus nerve, but they potentially connect For example, the vagus nerve, they connect with the GI tract. Potentially, it could even communicate with local cells like macrophages or mast cells or some of these other cell types that initially detect signals of nausea. So um, I would like to understand the signals, which cells detect it, and then we can actually target those cell types and block it at even the peripheral site before it reaches the brain. The sensation of nausea is always in the brain, but we can cut the signal transduction at different steps. Um, but we need to first understand what are those cells and what are those signals in different nausea conditions. For example, pregnancy or cancer chemotherapy or migraine. We really know too little.
Yeah, thank you for answering all those questions. I I know we went a little bit over the hour. You probably, it's late for you, so we don't want to take too much more of your time. So if anyone has a last question, please go ahead. So, yeah, go ahead, Victoria.
Yeah, thank you. Choo Choo, I really just would like to thank you because even if there are no definitive answers or solutions to nausea that people are suffering currently, it's meaningful to hear it acknowledged and see it acknowledged in this way. Denise, you reminded me when you asked if there was another method, a think method, to combat nausea, and I'm not criticizing your suggesting this because definitely so many things are possible. For example, stimulating the vagus nerve in the face of panic and anxiety is so effective and almost immediate. It can be, but I want to acknowledge that for women, many of women's symptoms that women are tradition or conventionally our complaints are ignored, and so it can be very challenging for a woman to be believed and find solutions. And one thing that I found that very prevalent was with morning sickness, with pregnancy, that Katarina said was so debilitating that she needed transfusions. I avoided the hospital by one blood test, um, and you're continuously told that it's in your head, that there's something that you could think and you could think it away, and it's not true. That it's real and you're being rational. But Katarina, you were mentioning the PTSD, if there was a possible link. There are two plants that I had in the house when I was first stricken with this morning sickness that lasted for months, um, and I cannot be in the presence of them without feeling nausea, and I will even forget that the connection is the pregnancy, and if I walk by them I'll feel instantly sick. And so I am so curious what we will learn about that connection, because all of those things, those are real. Those reactions are real. They have information that we can glean someday. And yes, my point is that it's important to honor what people are complaining of. It's important to honor the symptoms and how important it is just that your research exists, that is in itself honoring people's symptoms and suffering. So thank you.
Thank you.
Yeah, yeah, I can only agree to what Victoria said, and thank you, Tutu, for coming here today. And we wish you all the best and all the luck and all the funding for your future lab. I hope you get a wonderful offer, which you for sure will get because you did really elaborate, amazing work. And once you have your new position and you start new research or whenever you want, please come back. But would be honored to have you back. And yeah, thank you everyone for coming, asking questions, participating in the discussion. Um, And yeah, I hope everyone comes back again, and I hope you enjoyed it too. And thank you so much.
Yeah, thank you. It was fun. Hope to come back one day.
Okay, yeah, and then we can have a party for your new lab, wherever it is. We are, it doesn't matter. So we are always here. Okay, then, and yeah, if you like discussions like this, join the club, Science Society. We'll have more rooms like this in the future. We have our next room on Friday, and it's quantum physics room with Dr. VNS, who will talk about how a new discovery where they saw how electrons take fast and slow lane all at once, which is really interesting. And we'll have on Monday Dr. Lumba talking about cognitive immobility in a mobile world, which will be a really interesting room about um, yeah, cognitive immobility. I think it's really interesting. And then on Tuesday we'll have Dr. Gross and Dr. Follen talking about a magnificent mega bacterium he discovered, and about origins of life, uh, and we'll have many more rooms which I won't take up your time. Just follow the club and you'll see them. And yeah, thank you everyone again. Have a great night, morning, wherever you are, and I hear you all back soon. Thank you.
Thanks, everyone.
Okay, I'll close the room in 3, 2, 1. Bye everyone. Thank
Bye.
you.
Thank you.